Everyone, thank you so much for joining us. We'll be getting started momentarily. Good evening or good afternoon, wherever you are. Thank you so much for joining us tonight for Ask Me, our educational webinar series. My name's Audra Hines, Vice President of Care Services for the ALS Network. I want to thank our lead accessibility and inclusivity sponsor, Mitsubishi Tanabe Pharma America, as well as Maxim Healthcare and our mission benefactor families. The contributions from these individuals and organizations are really amazing, and programs like this one tonight, as well as the multitude of services we provide, wouldn't be possible without these generous donations. Many of you joining tonight may know the ALS Network well, and some of you might be new to our organization and the services we provide. Please know that if you or your loved one need a piece of equipment loaned to you, emotional support or counseling, assistance with finding home care, help understanding insurance benefits, guidance in finding an ALS specialist for care or a clinical trial, or just someone with expertise to answer any other questions about the disease, the ALS Network is here for you. Our committed staff is driven on a daily basis to provide high quality, personalized and local services, as we also fight on the legislative advocacy front and build our growing research program. To learn more about the ALS Network and the menu of services we provide, please visit alsnetwork.org for more information. Earlier this year, you may know we held a program like this one tonight to summarize the outcomes of the ALS Network's 14th annual California ALS Research Summit. On the East Coast, this past March in Baltimore, the 24th Packard Center Annual ALS Research Symposium took place, which is a gathering that allows ALS researchers to share their latest results, to fuel new avenues of investigation and foster novel collaboration between teams. We're so thrilled tonight to have Dr. Christine Vandevelde, Scientific Director of the Robert Packard Center for ALS Research at Johns Hopkins University. She's a professor in the Department of Neurosciences at the University of Montreal, and she obtained her PhD in biochemistry from the University of Manitoba and subsequently pursued postdoctoral studies at UC San Diego, where she developed an expertise in ALS. Her research interests are centered on understanding the underlying pathological mechanisms that lead to ALS and FTD. Dr. Vandevelde, thank you so much for being here. We're thrilled that you're with us tonight, and we're looking forward to your presentation.
Thank you so much, Audra. Really, I'm very excited to be here. And a big thank you to ALS Network for allowing this presentation. I'm really excited to share what happened earlier this year. Those of you on the call, forgive me in advance for my voice. I'm doing my my utmost. Are my slides? Slides? Yes, they are. Okay. Sorry, I couldn't see.
Yeah.
Couldn't see where they went. Okay. So thank you, everybody. Thank you so much. Really, really excited to be working with the ALS Network on this initiative with Packard Center. We believe strongly in getting the word out and collaboration, and this is just another extension of that, and with the opportunity to work with you guys. So thank you so much for hosting. I want to tell you guys what happened this year at the ALS meeting in Baltimore. But before that, I thought I would give a couple of slides to introduce the Packard Center for ALS Research to some of you, because it's something you haven't heard of before. So the Packard Center is a consortium of ALS researchers that is sort of centered at Johns Hopkins, but actually has a global reach. And the vision for the Packard Center is really about significantly altering the course of ALS and giving those people that are diagnosed with ALS the opportunity to live long and healthy lives. And we do that through a mission that is really focused around research, empowering collaborative breakthrough research that advances our fundamental understanding of ALS, and as well as supporting and engaging the global research community. And I'll showcase some of that in some of the highlights that I'll discuss with you. So the annual meeting is held every March in Baltimore, as Audra indicated. It's really a collection of global ALS researchers, Packard-funded PIs, their trainees from their labs, as well as select industry partners. There's also several partners from sorry, other sort of non-profits that are associated with the ALS cause that are also joining us at the meeting. The focus on the meeting is very much about networking and collaboration, and very much for the researchers to really show their unpublished data. The purpose is really to accelerate the sharing, the open sharing of data and resources so that we can get to answers faster. And so folks that are asked to present at the meeting, they're really encouraged that we want to see their latest and greatest unpublished work so that everybody in the room can benefit from this new knowledge and move the field forward faster. As indicated in the graphics, this meeting is quite large. We have over 300 people who come in person, representing 60 different institutions from 11 countries, again, speaking to that global collaborative mission of Packard. There were 43 talks this year, and accompanied by 80 poster presentations. And so poster presentations are usually led by the trainees in the lab, so the young students and postdocs, research associates that work with the lead investigators, and they're there to show their data firsthand of what they actually did. We were also joined by over 200 people online through a remote access program. We also incorporated 56 participants in a networking lunch in which young investigators and PIs were introduced to members of industry, from nonprofit, as well as leaders in the ALS community, both from the clinical side as well as in the fundamental research side, with the intent of trying to foster their careers in ALS and keeping them attached to our cause. And we had 13 sponsors of our events. Many of them are industry partners that I'm sure you're familiar with through various facets of the ALS community. They've been active partners in many different ways. So why does the Packard focus on fundamental biology? And the reason is simply this. We really want to take this healthy neuron and understand why this healthy neuron, this is a motor neuron from a human spinal cord that's been stained, labeled nice and brown. And why does it become these sick, dysfunctional neurons like these ones here, which might be observed in an ALS patient? And we believe that understanding how this process happens, understanding the fundamentals of this is what's going to guide the development of disease-modifying treatments and biomarkers. We need to understand this deeper biology and really be able to develop drugs and biomarkers that are really focusing on mechanisms and not necessarily on sort of unfounded principles, really mechanistically based. So I thought it'd be useful to sort of review a little bit about what it means to develop a drug. Excuse me. In general, there are 4 broad phases of developing a drug. The first is the research and development phase. This can be the preclinical research that I'm going to talk to you a little bit about. This can take on average 3 to 6 years, depending on the study or the target. Then the next is preclinical studies. So moving stuff, perhaps, for example, into a mouse model. And this is a little bit of an underestimate. It's really often more than 1 year. And then there's clinical trials, which of course, patients and their families are always very tuned into listening and hearing about, and these can take 4 to 7 years on average. And then there's the review and the regulatory approval process. And depending on where you are, also it has to do with that and access, the access to the drug. These are 4 broad steps. Underneath each one of these, there are many steps. It's very complex. In the R&D phase, you have to identify a target. You need to screen compounds to test it against the target. You need to identify, then refine those compounds into what was called a lead, but then we'll get moved forward into these preclinical studies. At the end of that, that's the compound then that would move in to patients in a phase 1, 2, or 3 trial. In general, these first 2 phases are often sort of done in academic labs. There are lots of industry partners that also do these portions of the work. but there's the lion's share often comes from academic labs. And this is a primary focus of the Packard Centers. They fund academic labs that are working in this early space to identify novel targets, both for drug development as well as biomarkers, so that we can track the efficacy of those drugs. How well do they work? How can we better stratify patients into different subsets so that maybe we might get a better answer in these clinical trials? So how do we do that? In general, the ALS field does that by trying to understand each of the pieces of the puzzle. And in this case, the puzzle pieces really are the individual genes. And as we know in ALS, there are over 40 genes that have been identified to date, and we're still identifying new ones. And in fact, we don't understand all of the genetics. We've filled in many, many pieces over the last few years, and there are many genes actually that should have populated that box and they didn't. Or we're seeing that we're starting to fill in the blanks. Or we're starting to see that these genes sort into different themes, whether it be looking at DNA and RNA. So the way that, the genetic material that makes each person unique, and how that material gets used into making the building blocks that make our cells and our neurons function. To mitochondrial dysfunction. So mitochondria being these specialized structures in a cell that is what essentially powers them and allows them to do all the things that they need to do. In the case of a neuron, allowing it to fire and so that you can move muscles. So these things are starting to sort into different themes. Excuse me. And so we structure a lot of the work at the fundamental levels around all of these themes. To understand ALS biology, I think it's really helpful to use this analogy of a puzzle. And so I borrowed this from Dave Taylor, a good friend of mine at ALS Canada, who has likened it to build these puzzle pieces. So in 1993, we didn't even have the puzzle out of the box yet. So in 1993, though, this was the discovery of the SOD1 gene. So now we knew where to look. And then in 1993, we discovered the gene. So now we got the pieces out of the box. We're starting to think about and work with this particular gene, developing different cell models, mouse models, fish models, fly models, all different ways in which we can understand the biology. And then in 2006, there was this discovery made about TDP43, which I'm sure many of you heard a lot about, a protein of significant interest in the field, and we'll talk a little bit about that later. We start to see that we're starting to assemble things, right? We're starting to get those edges around. When you build a puzzle, you always build those edge pieces first, and you start to fill in the center. And then in 2011, with the discovery of the C9 ORF72 repeat expansion, we're starting to get more and more of the puzzle pieces filled in. And each day we're getting a little bit closer, but we still haven't got the full picture yet. And that's why we really need to study the fundamental biology using different cell models and different animal models to understand what is going wrong. But if we understand what is going wrong in our neurons, then maybe we can figure out ways that we can fix them. So these puzzle pieces have contributed to many different mechanisms, as I alluded to earlier. And this is just the same mechanisms now grafted onto the neuron, if you will. And don't get lost in the details here. The point is just trying to show that the neurons are they're large cells. They're very complex. They have very specialized features. I mean, this very long axon that connects to the muscle to make the muscles move. And there are different, pathways here that affect the axons that might be primary drivers of the disease. There are other pathways that might be affecting these other extensions called the dendrites, how these neurons receive inputs from other neurons. There are these pathways that are happening within cells that regulate how the cells function as far as how do the proteins fold, how does the RNA get taken care of, all the things that a neuron needs in order to be able to function properly. And lastly, I would be remiss not to highlight that there are other cell types that are also important within our brains and spinal cord that contribute to how those neurons function. So for astrocytes, glia, microglia, oligodendrocytes, all of these contribute to that. Okay, so we're going to spend a few slides talking about some of the highlights from the meeting. I've broken it down into three main sections. I'm going to cover 3 vignettes from each of them. First one is about TDP43 biology. And so I'm going to ease you into that and explain a little bit about TDP43 and what it is and how it works. And then we're going to talk about some new technologies that were put on display at the meeting, as well as some new and I would say exciting findings somewhat even surprising that I think are cool new things to watch for the future. As a fundamental researcher myself, I'm excited about these to see how they develop. So TDP43, biology and cryptic splicing is going to be the first thing we're going to talk about. But let's talk about TDP43 first. TDP43 is a protein that normally lives within the nucleus of these motor neurons. So there's that cartoon that I showed you, motor neuron, there's the nucleus. That's the command center, if you will, of the motor neuron. And this motor neuron extends an axon that connects to the muscle, to muscle fibers, and controls the way those muscles function. TDP43, labeled in brown here, this is an actual human motor neuron taken from a spinal cord of an individual. This is where it's supposed to be. It's supposed to be primarily located in that command center. However, what we know is that the majority of ALS patients, it's not there. It's not where it's supposed to be. It's out in what we call the cytoplasm. So this is the other space in the cell. And it can adopt many different shapes. It can be these large circular things. It can be one large blob. It can be these fibrillar sort of things like a net. It can be kind of granular. And so there are some groups that are working at trying to understand whether or not these patterns mean something. And there's some interesting work that is coming out of Magda Polymedu's lab in Zurich that is definitely indicating that that might be true. But we see that one of the universal truths here is that we don't really see the nucleus, that the pool is not concentrated in the nucleus in that command center like it's supposed to. And that is thought to be important. So as I mentioned, this event happens in more than 97% of patients that are affected by ALS. And that doesn't mean that it happens in all the neurons at the same time. It means that it's happening in some portion of the neurons at any given time. So it's what we call stochastic. It's not uniform at the same time. It's like pop, 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 pop. This pathology, as it's referred to, is also observed in patients who have frontal temporal dementia. About half of patients who have frontal temporal dementia. And of course, we know that there are links between ALS and FTD that extend beyond just this. Whether or not the TDP43 being absent from the nucleus and now being accumulated in this other compartment in the cytoplasm, whether it means that we've lost some essential function in the nucleus, in that command center, or whether we gain some toxic function in the surrounding area in the cytoplasm, that's an area of really hot debate in the field. And there are studies that support both models, and there are even some studies that suggest it might be a bit of both. And this is a really important area in the field. Right now, I would say that there's been a shift in some ways, a focus on this loss of function, this idea that because TDP43 is not in the nucleus, maybe something is not happening the way it should. Now, when we think about what happens in that command center, what does TDP43 do there? Well, TDP43 does a lot of things in there. It takes care of the genetic material, the DNA that makes each of us unique. It gives us blue eyes and brown hair or dark skin. And all of those genes, they get made into something called RNA. These are sort of the messengers or the little soldiers that go out and actually they get used as a building block and made into something called proteins that do all of the biological functions that our neurons need. So when we think about it, I like to think about it that DNA is the cookbook, that big giant cookbook. And then RNA is actually that individual recipe, your favorite recipe for meatloaf or banana bread. And then what happens here is that the protein is actually the cake. It's the product of that, when all the pieces get put together. And this is the good part, right? We want the cake. This is the part that does most of the functions in a cell and allows it to do what it's to do. Excuse me. So we put that together now. I'm going to show you the first vignette from the Packard meeting. This is the work that's coming from Dr. Clotilde Agier-Torren from the University, from Mass General Hospital and Harvard Medical School. She has been working for many years on a target called Stathmin-2. So Stathmin-2 is a target that is regulated by TDP43. Now, ignore the right side of the screen for now. Focus here on the left on what happens in a healthy neuron. So I told you is that we have all this genetic material, our genes, and I'm sure you've heard about junk DNA, but we don't need all of the bits. And so when it gets made from DNA to RNA, there are bits that need to be cut out. These are called introns. Don't worry about that. These pieces, they got to get stitched together. And so that building block, again, this is the part that now gets made into the protein. So when TDP-43 is in the nucleus, this event, that stitching together of the functional bits, happens the way it's supposed to. And we get a functional protein called staphynin-2. Staphynin-2 is a protein that is really important to axons, that really long extension that the motor neuron uses to connect to the muscles and enable them to contract. In the context of diseases where TDP-43 now is not in the nucleus, it has left the nucleus, left that command center, that stitching together of events doesn't happen exactly the way it's supposed to. And in fact, this particular gene is quite unique because it's got an extra little bit that normally gets hidden when TDP-43 binds to it, and it doesn't get included into the final product. But when TDP-43 is absent, now it gets stitched in. And this is not a good thing because it interrupts the normal sequence, and now we don't get that functional protein that we need to maintain the axon. And so there's many groups that have been working on this biology for a while, including, I'm sure you've heard about clinical trials that are underway by Curalis, that is using antisense oligonucleotides that are going to try to hide that bit again and make it not visible to the splicing machinery. So that's in progress. But what Clotilda's been doing is doing a small molecule screen looking for compounds that might be able to regulate staphynin-2. And so this is work that's in early days, very early days, very interesting, because it might be a way to complement the ASO approach that is currently in phase 1 trial. Or could be even an alternative. Again, very early days, very exciting to see this work at a fundamental level happening in cells. And well, I'm excited to see what happens in the next few years as she moves this work forward, probably into mouse models, I'd anticipate. Okay, the second vignette I'm going to show you is from Pietro Frada. He's at the University of College in London, in the UK. So again, speaking to that international collaboration that is important to Packard. Another target of TDP-43 that is also cryptic splicing is UNC13A. So the exact same thing. You'll recognize the cartoons are very similar, but normally in a healthy neuron, there's a little bit that normally gets hidden away by TDP-43 when TDP-43 is there. And when TDP-43 is not in the nucleus, now that bit gets included. When that bit gets included, this is a gene that is important for synaptic vesicle. So when that neuron attaches to the muscle, the way that neuron communicates to the muscle is through the release of these synaptic vesicles, so little tiny bags of transmitter that tells the muscle, now you need to contract, now you need to stop contracting, okay? And so he's developing antisense oligonucleotides to target this cryptic exon, very similar to what's being done with staphynin-2. So he showed some very early data showing in cultured cells that there was rescue of these events. This is very exciting. Good preclinical data in human cell models, and now the work will be moving forward into mouse models. And I think that a phase 1 trial is anticipated. There's more than one industry partner that's interested in this target, so I'm excited to see this develop. Next one I would like to highlight to you is a effort from Michael Ward at the NIH, and it's premised on work that was first actually discovered by Phil Wong at Johns Hopkins. It is being supported by work by Len Petruccelli at the Mayo Clinic. All three of these are Packard funded investigators at some point in their career, as was Dr. Lagier, Toren, and Dr. Frada. In this case, what Michael has done, Dr. Ward has done, has shown that TDP-43 regulates many different events, many of these cryptic events in humans cells, and actually has even shown that they can lead to different outcomes. Sometimes it's going to lead to an outcome where the protein is going to get degraded, and in other cases, it's going to lead to an outcome where the protein doesn't get degraded. It actually gets made, and that it means it's a novel protein that didn't exist before. And so he's using this as an opportunity to cover novel biology, but proteins that normally wouldn't have been observed previously, but now become visible when TDP-43 leaves the nucleus. And he's also harnessing that information to see whether or not they might represent novel biomarkers. So that's being done in collaboration with Len Petrocelli as well. Phil Wong is also doing similar work. And this is wonderful that we have groups that are both collaborating together, but also sort of it's almost like a friendly competition, but it's 100% collaborative, because we love the idea that a finding would be independently verified by different teams using different sample sets. This is great. I know some people might be like, why are they repeating the same work? That's okay, because it gives us confidence that that's a real meaningful target. If more than one group identifies things in more than one set of patient samples, this is great news. This gives us way more confidence. And so the early work, this is a paper from Phil Wong's group that published earlier this year. They're proposing, actually their data showed that a cryptic exon in this particular target, HTGFL2, it's kind of like alphabet soup. Don't worry about what it means. This gene is actually showing this event, and it's occurring prior to the rise of neurofilament that is now becoming increasingly accepted as a marker of the axonal loss and motor neuron injury that occurs in ALS. So the idea that this might be something that could be detectable in a pre-symptomatic, so before patients manifest, before neurofilament levels start to rise, this is incredibly exciting. And I'm sure you can appreciate how that can be incredibly useful in various clinical trial settings. And so there's ongoing work to try to validate this now in larger sets. And I'm sure these three teams are going to take this forward in a meaningful way in the coming year. I need another drink. Okay. So the next piece I'm going to tell you about are some new technologies that were discussed at the meeting. I really see them as cool new tools to advance us, get us to our answers faster, get us to understand why are these neurons sensitive, why are they dying when they shouldn't be, how do we support them? Okay, so the first story I'm going to tell you about is from Dr. Claire Clellan. She is a new assistant professor, clinician scientist at UCSF. Very exciting work in which she's harnessing a new technology called CRISPR. CRISPR stands for clustered, regularly interspersed short palindromic repeats. Again, don't worry about the details. CRISPR is basically a pair of scissors. It is a specialized way in which one can go in and clip pieces of the DNA and cut things out or change them, correct things. She's harnessing this technology to show how this might be, oh my gosh, how this might be helpful to edit the C9 or 72 repeat expansion. Sorry, lots of noise upstairs. So this technology was originally identified in bacteria. This is actually a mechanism that bacteria use to prevent its infection by invading pathogens. This is a beautiful example of where fundamental biology in a completely unrelated field has got serious implications for how we extend our understanding of fundamental biology, but also how we might be able to harness that as a potential therapeutic. And so she's looking at how she can edit the C9 expansion in patients. And so here's a little snippet of her very recent paper. This was published, I think, the week after we met in March, in which she showed these here, focus on the pink arrows are showing some neurons in which TDP43, which is in green, is not in the nucleus. So the nucleus are these bright green dots. So here she's showing some neurons that are showing the TDP43 has left the nucleus. And these are C9 neurons. Okay, this is a patient that has neurons from cells that have an expansion in the C9 locus, and we see that with these different approaches, she actually can put TDP43 goes back to the nucleus just by correcting that C9 allele. This is very exciting. Again, very, very early preclinical. It is one of the main challenges of this technology is going to be how does this get delivered into the central nervous system, into our brain and spinal cord, in a way that it targets all of the cells that need to be targeted. Dr. Clellan is well aware of the challenges in the delivery aspect. And in fact, the entire second half of our lab is focused on that. I'm feeling very good that she's got full line of sight of how this might get one day adapted to use in patients. Again, early days, but exciting. The next I'm going to tell you about is another new person on the scene. This is Dr. Jenna Gregory from Aberdeen University in Scotland. She is a neuropathologist. She spends her days looking at tissue sections of patients. She is a clinician scientist. She is very passionate about her work as well. And she's designed a new way to look at TDP-43. What I showed you before, I showed you that image before, this was using antibodies. So this is a protein-based detection, going after the protein, using proteins to detect the protein. What she's identified is a new way that exploits the fact that TDP-43 usually binds RNA, those building blocks. So these are called aptamers. Again, don't need to worry about what they are. Just understand that these are a new technology that allows us to visualize things that we couldn't see before because the previous tools couldn't detect them. And so one of them, for example, is the idea of that there's pathology in a glial cell here. So often what we see is neurons, what I showed you earlier. But here she's showing that in a supporting cell or that's sitting next to a motor neuron, that there's also a mislocalization of TDP-43. This is very interesting, and we don't yet understand what the significance of that is. But I'm certain that this is going to spark entirely new lines of research and actually fill in that puzzle for us in a larger way, because the idea that these other cell types contribute to the disease is undoubtedly true. We've known that for many years based on early work from other teams using SOD1 mouse models, for example. So next I'm going to show you is about a study from Don Cleveland. So Dr. Cleveland is at UCST, San Diego. Full disclosure, he was my previous mentor, I realize. So small conflict, I suppose, but I'd be remiss not to share this with you because I thought it was very exciting work from his team. But he's adopting a new technology called MUFISH. So it stands for multiplexed error robust fluorescence in situ hybridization. Again, don't worry about the details. Just understand that this is a very powerful method that actually allows single cell resolution for gene expression. And it does that by maintaining the cell's spatial information. So here's a cross section of a mouse brain, and all these different colors are indications of individual genes that have hybridized in individual cells. And so you start to see patterns. So there's clusters, right? The pink goes with the pink. There's a cluster of green here and here, but there's also one here. And you also see the layers. This all reflects very well that we know that the way that the brain is assembled, if you will, both in mice and in humans, is in this very, very ordered fashion. And so this reflects that. This is very exciting because most of the traditional ways in which we looked at how different genes expression might have been changed in patient material or in mouse material is that we had to take the material out of the animal, for example, and mash it up. And so we lost all of that information about where did this cell or this gene come from? Did it come from here or here or here? And so now this puts this all together, and now you can really see it. And just to give you an idea, this is an exciting technology that you see is very much on the rise. You see that just in the matter of in 3 years, and there's now 40 papers, there are only 5. This is an 8-fold acceleration. I don't even know what it's going to look like in 2024. I've seen already a half a dozen papers that are using this method in different contexts and really characterizing how things are expressed in the brain. And this is important because we need to understand better where are the genes expressed and where are the cells that have gone wrong, and where are they the ones that have the problem? Understanding more detail. And so Dr. Cleveland is going to apply this technology to understand more about the C9 repeat expansion in the human spinal cords. And that's also another powerful portion of this technology, is that rather than going through mice, mice are a fabulous model and they do many, many great things for us. This allows us to look in humans. So new and exciting, 3 quick vignettes there. The first is from Dr. Peter Todd at the University of Michigan. So he reported on something called stress granules. So every cell in our body has different mechanisms that are engineered in order to allow them to survive different stress events. And so that stress event, I don't mean stress because your teenager didn't come home on time or broke curfew or didn't turn in their homework or, all the other stresses that come with life. It's really about sort of the viruses that we might encounter that we don't realize, or chemicals that we might encounter in the environment. All these different things. And so every cell has that ability to launch this mechanism called stress granules. And they're really, really important because they allow neurons, for example, to get through these stress, all our cells. So in the last few years, these structures had been seen to maybe be the precursors to all of that TDP-43 cytoplasmic aggregation that was observed, that I showed you, that brown stuff that was shown in those other images. And so there were lots of groups that were looking at ways to try to inhibit the formation of them as a way to prevent the formation of those TDP-43 inclusions, and maybe that might be helpful. And his data actually that he showed actually challenged that view and actually suggested that maybe they're actually protective. So this is some work in flies, where all you need to know is that this is a protein here that interrupts this way those stress granules form. And so you see that this is a fly eye. This is an assay that's used to determine whether something is toxic or not. And so we're seeing that we're able to restore that that structure. The eye looks more normal. It looks like the control when this tool is being used. So this could have implications for drug development efforts that are currently in progress, that are inhibiting this pathway, and also might encourage efforts that are looking to support this pathway. So new, exciting, early days. Definitely prompted a lot of discussion at the meeting, and I think that's noteworthy. Another talk that prompted a lot of discussion was work from Dr. Piero Passanelli at Thomas Jefferson University. Dr. Passanelli presented a novel therapeutic idea for ALS that is premised on work that has been pioneered in the cancer context, the cancer field. And it's the idea that a patient's own cells, T cells in this case, these are specialized cells that can be engineered to target the cancer cell and get rid of it. And so this is done by taking the cells from a patient. They're modified with something called a chimeric antigen receptor. This allows them to hone in on the bad cells, or the bad cancer cells. They're growing up, millions and millions of them in a lab, and then they're reinfused in the patient. When these cells get back in the patient, they seek and destroy those cancer cells. So she's looking to see whether or not she can do the same thing, but rather than honing in on cancer cells, we hone in on those toxic microglia that we know are there, because there's been years we've known they were there. And many of the ways that we've tried to target them in the past just haven't been successful because they weren't precise enough. So if this is successful, this could be really a novel therapeutic that would overcome those limitations. Again, this is very early stage research. This still needs to be verified using animal models first. So very, very early stage, but I think exciting, sparked a lot of conversation. This is really a novel out-of-the-box thinking. And this was the beauty of the Packard meeting, because there was so much dynamic discussion. It was great. The last thing I think I have for you is from Ruben van Eyck from the University Medical Center in Utrecht, which is in the Netherlands, in which he talked about really on a high level, how much does it cost to develop a drug? And I thought that would be useful to hear. It's a big number, $2.6 billion. And that our clinical trial success rate is really only 3%. And this is not for ALS, this is for all diseases, okay? And so he's really looking at ways to how to streamline this this normal trajectory of how we get to go through clinical trials through this phase 1 through phase 3. And he's doing that by leading an effort called the TRICALS, Treatment Research Initiative to Cure ALS. This is a consortium of 48 medical centers across 16 countries in Europe, in which they're trying to streamline clinical trials. And I think there's very important lessons there for us and for North America, and how we might do better. So he was invited to show off what is being done in Europe, so that we can learn from each other. Again, cross-pollination of ideas across the Atlantic, super important. And I do want to emphasize here too that just to keep in mind that this is really important, that if I cover that early part, that drug identification, construct, screening, that's that preclinical, they don't have anything to put in the pipe. So all that stuff I showed you before is to feed this pipeline. Oh, so the key takeaways that I want you to have are that therapies and biomarkers should be founded in understanding of the biology of the disease. Research is really a collaborative global effort, and the Packard is 100% committed to that. and investment in preclinical lab-based studies are really clinical to drug and biomarker discovery. And the Packard does that by facilitating catalyst funding for many of these investigators so that they can advance their work and leverage it into larger funding. We can get them advanced into those clinical. If you'd like more information, I'm happy to answer questions, but also please check out the website at thepackardcenter.org. Happy to take any questions, Audra, if there are any.
Thank you so much, Dr. Vandeveld. Oh my gosh, I learned a ton. I love how you did the 3 different vignettes in the 3 different buckets. That was really interesting and just kind of a great overview of the whole conference. Much appreciated. A few questions that were submitted ahead of time and a couple questions we have now. And just for everyone in the audience, if you do have other questions for Dr. Vandeveld, just type them into that Q&A function at the bottom of your screen and we'll get to as many as we can. Wondering, we've actually had a couple of questions submitted by a couple different people ahead of time asking about Dr. Michael Strong's research and his I think maybe upcoming trial. If you know anything about that, or just maybe your thoughts on that, if you don't mind answering that, that'd be great.
Sure. So yeah, Michael Strong is a Canadian researcher at the University, Western University. He's published a new finding in which he's reporting on a small piece. So for many years he's been working on how TDP-43 interacts with a protein called RGNEF, which is another RNA binding protein. He has now refined that interaction to a small sequence or a peptide called NF242. What he's showing is that TDP-43 and NF242, they bind, and he's proposing that when they bind, that this seems to limit the toxicity that TDP-43 evokes. He's shown that using some fly modeling, as well as in some mice, to say that it's very early stage research. The work is interesting. I think that we have to be cautious about the interpretation of things as usual whenever I'm always cautious about things that are demonstrated in models because I think we have to really wait till we get to those clinical trials in patients. As far as I understand, I think that Dr. Strong is committed to moving this forward into a clinical trial. I think he's a little ways away from that because, as even stated in the paper, he's going to need to refine that small peptide that he's generated to get it as small as possible so that it doesn't invoke any negative consequences in the cells. So interesting work for sure. Early days.
Okay, thank you. That's really helpful. Another question someone just chatted in, would CRISPR DNA editing therapy be limited to only the 10% or so of ALS patients who developed ALS symptoms, based on a genetic type?
So yes and no. Okay. So the original, the primary target that Dr. Clellan is looking at is C9orf72, which of course is also found in a good proportion of patients that we classify as sporadic, not having a family history. So it would also be applicable to them as well as those patients that would have a family history of C9. Again, that work is still very early. It's a new technology that's being adapted into the clinic. That technology has been implemented in a clinical trial for disorders affecting the blood. To keep that in mind though, the blood is an easily accessible tissue, if you will, right? You can get blood from a patient, fix it, put it back in, that's okay. When we're talking about trying to change things within the brain, within the nervous system and spinal cord, delivering that machinery, those pair of scissors, is a little bit more difficult. But again, Dr. Clellan is working on strategies for that in parallel to the work looking at the editing.
Okay, thanks so much. Question we had submitted ahead of time, wondering if there are any research or treatments that you know of in the pipeline that are specifically for other motor neuron diseases, PLS, things kind of in that category. Any trials or research happening around those specifically?
I think the one that I've seen quite a bit, in social media, which I'm sure many attendees have as well, is from Northwestern University, something that was called New9. the press releases were a little overstated, in my opinion. And again, that's my own personal opinion. But there was, I think, there could be some promise in that when it comes to primary lateral sclerosis, the PLS, because I think that some of the primary data that was shown really seemed to show a rescue for upper motor neurons. I do believe that the lead PI on there, Dr. Silverman and Dr. Hande Osdindler, is moving that work forward at their institution.
Okay, terrific. That's great to understand. Any progress in antisense treatment for those with C9orf gene? You know,
right. So, of course, we're all very disappointed about the news earlier this year from Biogen and Wave that they discontinued the C9orf72 ASOs. But I'd have to say I want to be careful. Because it didn't work, it doesn't mean it failed, because an experiment was still done. Like it or not, it was a phase 1. It was an experiment. And I'm grateful to patients that agree to participate in these things. But that trial was actually informative because it told us very quickly and very conclusively we were barking up the wrong tree, right? Because the neurofilament signal was so clear. And so it immediately prompted the community, and that was also something that was heavily discussed at the Packard meeting, because the announcement came out 3 days before our meeting, 2 days before our meeting, and so everybody was discussing it. That was the conversation at all of the coffee breaks and at the posters, and in between sessions, people were talking about that outcome. And so I'm happy to report that there was so much dynamic discussion. People were immediately thinking about, what did this tell us? And so one of the things it told us is that maybe we targeted the wrong strand. So we know that the C9orf72, they're both strands, but what we call the sense strand and the antisense strand. I know that's confusing, but it's not the same thing as the antisense oligonucleotide. The DNA has 2 strands, it's the sense and the antisense. And so all those trials that were done were targeting the sense strand. And so folks are immediately thinking about whether or not they should be targeting the other one. And there is evidence, actually, when you comb the literature from cell models and mostly from human cell models, that other strand might have been doing something. So I think there are many good teams that are working on it. Jeffrey Rothstein, actually, Dr. Rothstein at Hopkins is one of those groups, and there are many others that are equally working on that.
Awesome. Thank you so much. Interesting question, and this might be tricky to answer. Someone's asking, how will the $1 billion in bipartisan ALS research that was recently approved, how does or doesn't that impact the FDA review and approval process and protocols? I suppose wondering if funding helps move that along, or if you have any opinions.
That's a great question, but I'm a firm believer that if I don't know, I say I don't know. Number one rule of a good scientist is, hey, you don't know when you don't know. It's a great question that I think I would need to dig in a little bit more of what the impact of that's going to be. I think it's great that there's going to be this investment, and how that, I don't know the answer to that yet.
Yeah, that's hard to know. Hopefully that will reveal itself. I don't see any other questions in the Q&A function. If anyone else has anything, please go ahead and chat that in. I have just one more thing to ask you, Dr. Vandeveld. This is everything that I've seen tonight, and I'm sure everyone else who's on the call is thinking this too. So much of this offers hope, right, for people living with this disease every day and their family members. What are the 1 or 2 things for you that keep you really inspired in this work and really keep the hope alive for you, right, in helping hopefully cure ALS someday?
Oh, so probably one of the things that I get, probably a couple of things. One, so I've been working on ALS since my postdoc. So it's been a little over 20 years since I first entered the field. And just for everybody on the call, I was actually a cancer researcher originally. And I came to ALS because I really wanted to understand a situation where cells were dying when they shouldn't. Whereas when I think about cancer, I think about cells didn't die when they should have because they get a tumor. And what I was struck by was and it's still true today. I think it is that how committed folks are to the cause. The researchers that are involved are really, really dedicated, and it's a real close-knit community, really focused on collaboration. And I wouldn't say that all fields have that, and ALS definitely has that. When I talk about collaboration in the ALS space, I mean, I don't think all fields have that. I think we're incredibly lucky. That gives me a lot of hope, because I think it's by working together, by sharing information rapidly before publication, which is what we do at the Packard Center. This is really going to drive the field forward. In that, I would also say one of the things I'm very excited about is to see how the field has become populated over the years, where how the field has grown in the both in the knowledge of what we and how we understand, how many different genes, how many different models, how many different groups that are working on it, and how they come, actually they've sort of been populated from other groups. So groups like Dr. Cleveland has seeded many of his people have gone on to be independent investigators. Same is true for many of the other people that I've talked about. And whether they're academic labs or they're in industry, that's another thing that has changed a lot in the last 20 years, is how industry has become a real partner with academics. You see a lot of cross-pollination, and there's none of this, well, that's mine and that's yours, and don't show industry because they might steal it. Honestly, this doesn't happen in the ALS space. Like, I think a lot of people kind of think that it does. It's just that sort of that conspiracy theory, if you will. There's really, I mean, we have academic partners in the room at the Packard meeting. There are tons of meetings where they are present. They are active partners. We talk and share our preclinical work. Do we sign some paperwork before sometimes? Sometimes, yes. Okay, because our institutions really need us to do, the academic institutions want us to do. But there's really the driving forces about collaboration, both between academic labs as well as with industry. The other thing I think is that the third thing I could say that I'm really excited about is that the time it takes from discovery to actually getting moving to a trial is getting shorter and shorter. Mm-hmm. And so to think about the success that we've experienced in the community for SOD1, right, with Cal Saudi. And so just for those of you on the call, I was a postdoc in the Cleveland lab when that idea was first launched. And I am thrilled to have witnessed in my career a drug that went from the early preclinical, that idea, to testing in rats, to testing in mice, to testing in monkeys, to testing in the phase 1 trial, to back up when they had to redo things, and to see a drug approved. Mm-hmm. I don't know that a lot of people had a chance to say that in their career. And not just a drug approved, but a real one that really changes the landscape for families with SOD1. And so, and that, all of that pioneering work is what allowed the Stathman 2 ASO to go to trial within just a handful of years. The same for the C9, it went to trial. And while that didn't work the way we had hoped, it still went to trial super fast. We have Jessifusin, which is targeting FUS, is in clinical trial. All of that, the acceleration, that the shortening of that timeline between discovery and getting it into patients and getting answers, that makes me very excited for the field.
Oh, I love that answer. Thank you. That's great. Okay, I've got a very sciencey question for you, Dr. Vanderbilt. Mm-hmm. Okay, is ectopic TDP43 glial pathology always associated with neuronal TDP43 pathology, or is it independent?
Great question. And I think the answer is we don't know yet. Because so it had been reported very, very rarely that using the antibody-based tools that we were always using, that there was TDP pathology in glial cells, but it was really not very often. And so with that new technology that's been described by Dr. Jenna Gregory, I think that we're going to start to see a more robust investigation of that, because her tools really revealed it a lot easier than what we'd seen with the previous set of tools. So I don't think we don't quite know the answer to that. I would definitely stay tuned. I think that's difficult.
Okay. Couple more questions. Is there any additional research on the effect of chemotherapy on ALS?
I don't know the answer to that. I wasn't familiar that chemotherapy was being used in the context of ALS.
Or maybe somebody who has ALS and they are on chemotherapy, right, to treat another condition, just if you happen to know.
Yeah, no, I'm sorry. I'm not familiar.
Okay. Okay, we can try to get an answer later for that person too, so no problem.
Yeah, okay.
Last question, which of the newer technologies or treatments seem to present the most promise? You talked about this a little bit, in your opinion. You did talk about this a little bit
Oh,
already.
well, that's a loaded question. That's like asking me to pick my favorite child. I can't do that. I think that all the things that I shared with you, I think are really powerful and are going to be really important contributors to getting us to understanding the root cause. I think, I think it's important that we bet on one horse because we don't know which one is going to win the race. And in fact, we might need more than one. So
That's right.
I can't answer that, I think, the way the person would like me to. I'm sorry.
No, that's great. You have been tremendous. This was one of our best webinars. I can't thank you enough for being here, Dr. Van de Velde. Really appreciate your time and energy in preparing this for us tonight. I think we have just a few other slides we're just going to wrap up with. I just want to thank everyone so much for joining us tonight, everybody who's been here. We're really grateful to each of you for your time and commitment, being a part of our community. A big thanks to all of our sponsors for their support. And if you have other questions that weren't answered tonight, including questions about how the ALS Network is involved in scientific research and legislative advocacy to support that, please do email us at askme@alsnetwork.org. We also invite you to save a couple of dates coming soon. This Sunday, June 2nd, is Lou Gehrig Day. The ALS Network will join with our partners in Major League Baseball to honor the legacy of Lou Gehrig and celebrate the ALS community. For any questions, please contact us at baseball@alsnetwork.org. And please join us for our next Ask Me webinar, like the one tonight, which will be held on Tuesday, June 25th, which is an update on what's new in speech and communication technology with Amy Ramon, who's a speech language pathologist with Forbes Norris ALS Clinic in San Francisco. We also invite you to sign up for one or more of our community events this summer and fall. Please do join us for one or more of these inspiring events. Love to see you at any of these. And finally, as you know, philanthropy fuels everything we do, and we're grateful to committed donors who give generously throughout the year. To learn more about how you can support the valuable programs that we offer, like this one tonight, please visit our website at give.alsnetwork.org. And please watch for the short survey on tonight's presentation that you'll receive via email to help us plan for future educational programming. Thanks again, and we'll see you next time.
Thanks so much.
Thank you.